Hello, everyone. Thank you so much for joining us today for our inaugural Spring into STEM session for medical physics and biomedical engineering. Delighted to have Ileana with us today giving this lecture. So without further ado, I'm going to hand over to Ileana to introduce herself and take things away. Over to you, Ileana.
Thank you very much, Naomi. Good afternoon. As Naomi said, my name is Ileana. I am a research scientist at MPL and currently in the fourth year of a part-time PhD with UCL. I would like to introduce you to my research in support to the radiation biology community that is currently performing preclinical irradiations. And with that objective, we have designed an end-to-end dosimetry verification test. To get us into the field of this work, I would like to introduce you to the pathway of the translational research, as well as to the place of preclinical irradiations within this pathway. Research results in this area are very important because they translate into new cancer treatment strategies, and specifically those involving radiotherapy. Preclinical irradiations are performed in so-called image-guided small animal irradiators. These are complex machines and devices that mimic clinical linear accelerators, except for that they have smaller dimensions and the energy of the X-ray beams is in the kilovoltage range. But what it is an end-to-end QA test? It could be defined as an audit or a dose verification methodology aiming to confirm that all of the steps involved in the irradiation process are performed in such a way that the desired radiation is delivered accurately and only to the intended region of interest. As a norm, results of this test are reported to each individual center participating, but are anonymized for publishing purposes. They have to be willing to participate, but the results are known only to them. With that in mind, and following previous experience of end-to-end tests designed to accredit radiotherapy centers for human clinical trials, we selected a mouse and a petri dish-like phantoms to mimic the most commonly irradiated specimens in preclinical research. So you can see here our mouse phantom, our petri dish phantom. But when dealing with irradiations, as we probably know, the shape of the phantom is not the only important feature to consider. We have to also consider the material composition, which is very important as the interaction of particles with the matter. And therefore, the dose deposit will be highly influenced by the type of material of tissue. So in this work, we consider three main mouse tissue types that were bone, long, and soft tissue like muscle. And the attenuation characteristics of each of those materials were experimentally verified in this type of setup at MPL. Subsequently, the mouse phantom was created by 3D printing the mouse skeleton, as you can see here, by milling the lungs and assembling them within the bone structure, and finally by casting the body shaped with the soft tissue-like material. The petri dish phantom was made of soft tissue-like material, a very simple phantom in this case, and with homogeneous material at all. But then we have already talked about the phantom, but every end-to-end test has a phantom and a detector. So now is the time to speak about the detector. Why did we choose alanine? Alanine is an amino acid that responds to irradiation by creating free radicals. The relative concentration of those radicals can be determined by electron paramagnetic resonance spectrometry, commonly known as EPR. This free radical, the concentration of those free radicals is proportional to those over a wide range. And the radicals are relatively stable, and they have a small temperature dependence. And those are all very positive features. However, alanine has an energy-dependent response in the kilovoltage range, and that needs an investigation. So energy dependence of detectors is, as we can imagine, not a desirable feature or characteristic. However, it can be evaluated and taken into consideration at the time of reporting results. So to determine the energy dependence, a measurement configuration shown here close to that of the intended use of the detector in the MICE phantom was created. Alanine pellets were exposed in a special plate of WT1 designed by us and built, customized to us. Those alanine pellets were exposed to unknown, untraceable values of dose at 4 different medium energy beam qualities, shown here in this table. And as a result of this experimental characterization, a quality-dependent correction factor to the reported dose at the quality in which these alanine detectors are calibrated, which in this case is cobalt, was found. And this factor is shown here, and the fitting to that performance or dependence is shown in that graph. And it's important to know that in our work, we are reporting quality of medium energy beams in terms of their half value layer or HVL in millimeters of copper. So at this point that we have a phantom, a well-characterized detector, and a curve that describes the response of that detector to different qualities. So our next step was to validate the experimental results with Monte Carlo simulations. We did just that, but by comparing the ratio of the absorbed dose to the alanine pellet inside the mouse phantom to the alanine pellet in so-called reference conditions. And the Monte Carlo ratio was 1.13 with certain uncertainty, and the experimental radio was 1.13 as well with a different lower uncertainty. So that is a good for us result, showing that we can carry on and then define or design a workflow for our end-to-end test or audit. And that's exactly what we did. We designed a workflow for our end-to-end test that comprises of 4 main steps. And then now, so the first step, it was the positioning of the phantom for imaging and segmentation into the different tissues of the phantom. Subsequently was to irradiate the pellets, and then you can read all the different steps that pellet irradiation part of the workflow consists of. Then gathering information for offline processing. And finally, those calculations, comparison with measurements, and reporting. So 6 institutions across the UK, you can see in the map where they were, the different institutions accepted to take part on the first round of our end-to-end test. This is the mouse highest position in the irradiation device. Sorry, I got my, okay. And in this, we can see there are different irradiation configurations that we use at each of the visited institutions, and those configurations were selected according to what was their routine irradiation practices. It could be a single anterior field, or it could be an R or 2 opposed fields. Those were the configurations we used in each of the 6 institutions that participated in this end-to-end test for the first time. So as a result of this work, in total, 62 alanine pellets were analyzed. And this is just an illustration for you to see all of the pellets and the results that were measured with each of them, but it's just, it's nothing you can get from it, just to give you an idea. But after the analysis, the first result that we came out was that the contouring process of the pellets that defines the volume of the pellet actually does not have a significant effect on the mean calculated value of those. And that's an important result for us. Then we also found that in the specific case of multi-plant treatment planning system, the segmentation process is crucial, and that a bad segmentation could lead to erroneous value of the calculated mean dose. And both this and the previous result are important to the comparison with the measured values of those, in the alanine detector. One of the most important results was that the end-to-end test demonstrated that errors on the TPS commissioning files could be spotted. And see here, a 14% difference between calculated and measured that was measured at institution number 1. So after we saw that result, we have a conversation with the manufacturer and they recommissioned the machine. And when we repeated the end-to-end test, the difference between the calculated and measured dose went down by 10%. So they are now at the level of 4%. So that was a very good result to see that we can spot errors in the commissioning process in this case. Another kind of unexpected result was shown when comparing phantom's complexity. In that sense, results are slightly better when pellets, can you see here, when pellets are irradiated in the homogeneous Petri dish phantom against when they are irradiated in the mouse phantom. And this is a graph that summarizes all of our results together. We can see the difference between calculated and measured dose for 80% of the evaluated end-to-end test are within plus minus 5%, and 100% of the end-to-end test performed are within plus minus 10%, which for preclinical dosimetry in medium energy X-rays is a very good outcome, and one that reinforces the confidence in the targeted preclinical irradiations that are being performed. The graph, in our opinion, also offers a picture at a national level of the accuracy of the delivered dose to preclinical samples as a representation of the institutions that are performing that type of irradiations. So as a summary and final remarks, this end-to-end test was implemented on SARP from Extral manufacturer small animal irradiators. However, the methodology is applicable to any image guide irradiation platform. The end-to-end test proved to be effective at detecting errors at the time of TPS commissioning. At one institution, we managed to spot that. The measurement results are traceable to the AUK Air Kerma primary standard and also to the recognizable expertise of the MPL Alanine service. The results derived from the proposed standardized methodology offer an overview on the achieved accuracy of values of those delivered by preclinical irradiation techniques across the different radiobiology labs. And implementing this type of tests allows to establish national records of the stability of the dosimetry performance. And this is a similar approach to what it is routine in clinical radiotherapy in the UK, without this in reference and non-reference conditions or for accreditation to participate in clinical trials. So this is, we are trying to get that across, the importance of doing this type of test. Originally, this end-to-end test was based on in-person visits. However, a postal modality is also feasible. We have the procedures in place to perform this in a postal modality or in a postal way. So with this, I would like to acknowledge all of the people that made possible this work. First, my colleagues that's from UCL and MPL that supported the work directly. Also, a UK Univate grant that was at the time a big help. Also, the members of the institution participated that gave their time during the irradiations and provided us with all the data so that we could analyze it. And to people at MPL that are responsible for reading and analyzing the alanine pellets for this study. There is a little bit, something little here that is a link to the paper that was published with the result of this work. But I don't want to finish before telling you that we are working forward to, we are thinking of other work that we are going to do, and we are already started to do in support of the biological, radiobiological community. And in this case, we are aiming to create a 3D printed mouse that combines alanine and films, and to implement the end-to-end test methodology for X-ray cabinets, where targeting is needed, but image guidance is not available, unfortunately. And also, we will be soon commissioning a very small plastic scintillator that could be later positioned inside a mouse phantom and will allow for instantaneous and in vivo dosimetry. Because as I didn't say that, but alanine is a detector that you have to read after. It can take maybe 1 week to get the results after you perform the irradiation. So with that, I will go back to the acknowledgement slide, and I will be happy to answer any of the questions that you will have. And thank you for your attention anyway. Yes.
Thank you so much, Ileana. If anyone has any questions, please do pop them in the chat box or the Q&A, or if you raise your hand, we can unmute you as well. And while we're waiting for some questions, I've just got a couple for you, Ileana. As the talk today was about the preclinical research and irradiations in small animals and cells, why is the dose verification so important?
They are very important for, firstly, because all the research that is performed in these small animal irradiators with small animals or cells is going to be translated into clinical trials. And there is the response, the dose response curves are very steep curves. So any small error in the dose that is delivered, it could translate in falsifying the endpoints that this preclinical research wants to achieve. So that is something that there is still a work in process for the radiobiology community, but mainly not only for the radiobiological community, but for the decision makers, in understanding that within the budget of the preclinical research, there is need to include budget for preclinical dose verification. So that is why the work that we are trying to perform is important from that point of view.
Fantastic, thank you. And then is it possible to use other types of detectors in this kind of end-to-end test?
It is possible in a way when it's about to, for instance, determine those in more like reference conditions where there are not collimators moving around or gantries moving around. Yes, you could use ionization chambers, but in these small animal irradiators, they are very contained. They are very enclosure, so it's difficult to have a cable moving around that all of the rest of the part of the device will be moving. So the alanine offers that possibility that is a cable-less kind of detectors. So in the future, we are trying to introduce detectors that could be read out instantaneously, like this small scintillator that I mentioned at the end. But the challenge will be exactly that, how to place the cables on the device that makes the readout so that it doesn't interfere within the movement of the device.
Fantastic, thank you very much. I'm just looking in the chat and the Q&A. There doesn't seem to be any questions. So I think that's a fantastic place to end the lecture today. Thank you so much, Ileana, for such a fascinating and insightful lecture. Thank you everybody for joining us today for our first Spring into STEM lecture. We are running these lectures every week throughout May, so do check out our website for more of the sessions and sign up to join those. And we hope you all have a lovely weekend ahead. Thank you so much, Ileana.
Thank you very much for inviting me. And thank to everyone for participating. Bye-bye.
Bye. Bye, everyone.